Hi, I'm Dr. Kevin Winthrop. I'm a professor of infectious diseases and epidemiology, public health here at Oregon Health Science University in Portland, Oregon. It's a pleasure to be here.
And I'm Dr. Richard Conway. I'm an associate professor at Trinity College Dublin in Ireland and a rheumatologist at St James's Hospital in Dublin. And yeah, delighted to be here as well.
Richard, you and I are going to wax for a couple minutes here on the significance of the recent oral surveillance trial. Before I ask you, how it's changed your practice and what you're seeing in Europe, I'll quickly summarize the findings. This was an FDA mandated study, as everyone knows, based off of the original approval pathway from the development program in RA, where there were some safety findings seen at the unapproved dose of 10 milligram BID of tofacitinib. So FDA made Pfizer perform this randomized controlled trial comparing tofacitinib at two doses, both the 5, which was approved, and 10, which was the unapproved dose, with a TNF blocker. And this was a trial primarily powered and created to look at the issues of infection, malignancy, and cardiovascular events. And so, actually, patients with cardiovascular risk factors over the age of 50 were selected for this trial. And the trial was a non inferiority trial based on the malignancy and the MACE, or the major adverse cardiovascular events outcomes. And the trial was continued until a certain number of outcomes were hit in each of those buckets. And what we found, just to summarize, is that from an infection standpoint, really nothing surprising. Everything we found was similar to what we found in the tofacitinib development program and the development programs and other real world data associated with the other JAK inhibitors. And that JAK inhibitors, or in this case tofacitinib, seem to carry the same risk of serious infection as the TNF blockers under study in this program, which were adalimumab and etanercept. Depending on where you were in the world, you were enrolled to one or the other if you weren't on the TOFA arm. So, with regards to infection, we saw this increased risk that was about threefold higher with TOFA for herpes zoster, and that's a known risk factor. That's similar to what we've seen in other studies with the other JAK inhibitors. And at the 5 milligram approved dose, we saw really no difference in serious infectious events. So, no surprise there. I think where the surprise was particularly with malignancy and then maybe also the MACE events. So MACE events were TOFA failed to be non-inferior. The relative risk was higher with TOFA, and for malignancy, the relative risk was higher with TOFA at both doses. And I think that was somewhat surprising. At least it was to me, because at least all the data we've seen, with all the JAKs so far, suggests there's no increased risk of malignancy as compared to TNF blockers. So I think important to remember that the comparison is TNF blockers. It's not nothing. It's not another compound, but it is TNF blockers. TNF blockers might at least be protective against MACE. I think they are. I think there's probably widespread agreement that they are. But with regards to malignancy, again, I think that was my most surprising finding. I'll just say that to summarize those findings, those were primarily an increased risk in lung cancer and somewhat in lymphoma, and it was driven by patients aged over 65, particularly those with smoking histories. Those were the primary risk factors, for the patients who developed malignancies in the trial. So, with that as summary, we're going to talk about what EMA and FDA decided to do about this later. But Rich, what are your thoughts? What are you seeing over there in terms of your practice and those around you?
Yeah, it's interesting because it's the JAK inhibitors were quite slow uptake even before oral surveillance in Ireland for a variety of reasons. We only had really two on the market, the non-selective JAKs and tofacitinib and baricitinib. And to some extent, people seem to be kind of waiting for the more selective agents to come along. Um, and then we had other issues both with reimbursement and prices because TNF inhibitors are substantially cheaper in biosimilar form in Ireland than JAK inhibitors. And also we had issues with shingles because we only got Shingrix vaccine into Ireland this year. So there were some concerns around that. But I think certainly in terms of where we thought we might be at the moment with JAK inhibitors, I think this study has given people pause. Physicians have been somewhat slightly more reluctant to prescribe JAK inhibitors than we thought we might be. Um, this kind of idea that there was going to be a revolution and JAK inhibitors were going to replace all these injection drugs and we are only going to have patients on oral agents. It might have been unrealistic anyway, but I think it certainly hasn't happened in part due to this. I think the big pushback I have seen has actually been from the patients. And even though we can talk about how these are relatively low risks and it's only compared to the TNF inhibitor, I think cancer, especially, and cardiovascular disease really frightens patients. And I think there have been a number of patients I've discussed it with, and they've just said, no, I'm not doing this. I don't want to accept those risks. I don't know how things are in the States, but
Yeah, it's interesting. I mean, I should also mention the VTE issue, the venous thromboembolism issue. That was, that's been on everyone's mind ever since the baricitinib program, where this was, identified as a potential risk. I think the data from this trial, particularly the 10 milligram tofa dose, the unapproved dose, I mean, there was an increased risk in VTE at that dose. There was not at the 5 milligram approved dose. So that was relatively reassuring, I think, for people using the approved dose. But the fact that there was a signal at the higher dose does make you believe that there is probably an actual an actual risk here to JAK inhibition. So it made me believe that maybe the Berry data is real. Maybe that potential risk flagged by FDA is actually real. So I don't know how yet, and I don't know that anyone has figured out why and what the biologic plausibility is behind that. But I think what was seen with the oral surveillance program does make you it does give you pause, and it does make you probably want to steer JAKs away from people, at least who've had prior events or DVT events, who are not anticoagulated, for example. I don't know, has that entered into your treating framework there at all in Dublin or?
yeah, no, I think so. I think there's always this consideration of trying to steer patients away, and we've been perhaps slightly even more reluctant to give it to older patients that have high cardiovascular risk, people who have high thromboembolic risk. Um, then what worries me a little bit is about, especially with the cancer stuff, you were saying there's this increased risk of cancer, which was 55 patients treated over 5 years to develop one cancer, and you're giving it to young patients who are potentially going to be on the drug for 20, 30, 40 years if it works. That length of time exposure really does worry me. So, yes, absolutely, it has. I have steered away from these kind of higher risk patients to some extent, but I am a little bit worried about the patients we wouldn't consider as high risk and the patients who weren't the included population in oral surveillance as well.
Yeah. I mean, that's a good point. We don't know a whole lot about people under 50 without cardiovascular risk factors, right? Those people weren't in that trial. Of course, what we know from the developmental program data and from other real world data, registry data, whatnot, what we've seen is very reassuring in those populations. Um, so, one might speculate that if you did a trial in those groups that you'd see reassuring data, but we just don't know. I think this was kind of surprising with oral surveillance, but it did highlight there are subgroups of people that are at are higher risk. So I do think rheumatologists can use this data to help steer the drug where the benefit risk ratio is the greatest, which is what you guys already do, right? I mean that's what you're always doing, and shared decision making with the patient and trying to explain these things. But I think the data from that trial, if you're 50 years old and you have one cardiovascular risk factor, you're probably not really at increased risk for some of these events we're talking about. But certainly, the older you were, the more increased risk it was, particularly with this malignancy signal. I don't know. I mean, I think it certainly raises the need for further studies. And to sort out whether these things are real and what's the reason behind them. I think we're getting there. I don't know. Do you think all JAKs are equal, Richard? I mean, that's kind of what's come out of this, at least with the FDA guidance, right? That the better safe than sorry approach that, hey, we saw this with TOFA, we're going to see this with everything.
I think that has to be the default position. We have to presume it's a class effect until somebody shows us otherwise. and the manufacturers may come to us and argue about selectivity and everything else, and that maybe their agent doesn't do it. But I think if people really believed that, they would be doing studies. So we have the TOFA study, we have a Barry study because they were mandated. We don't have
Yeah.
a upadacitinib, we don't have a filgotinib study. And I think if the manufacturers of those drugs were fully confident in their agents, they'd be running that study themselves to prove it. So that
Yeah.
worries me a little. Um, yeah, I think the onus has to be on them to prove it to us, for the agents to prove themselves to be safe before we try and justify it with any of this selectivity
Yeah, I mean,
issue.
the FDA's guidance, I think, was certainly conservative. EMA was less conservative, right? They just kind of said, hey, put TOFA behind TNF blockers in certain groups of people, patients over 65 with risk factors. I think the FDA was more like put all the JAKs behind TNF blockers in everybody. I don't even know if they issued an age group. I don't think they did. I'd have to check, but certainly a more sweeping recommendation. But I don't know. There is a selectivity issues, but there's a lot of differences. Like NK cell behavior is different with TOFA than it is with Filgo or UPA. So one could speculate there could be differential signals, particularly with regards to malignancy and some of the other events on the study, based on the fact these drugs are different. I mean, the other thing too I'd point to is the dosing, right? I mean, I think as all these drugs get pushed up, they become more pan-JAK. And TOFA at 10 is a lot more pan-JAK than TOFA at 5, and I think that might help explain why some of these adverse events were more common at the 10 dose than they were at the 5. So I guess from a rheumatology standpoint, I think it reiterates that the FDA's decision initially was right to approve the 5 dose and not the 10. And then I think further refinement of who to use the 5 in is justified based on the current data. So I don't know. Anything else to add about how this makes you feel, Richard?
I suppose the other thing we always have to remember is with this trial, compared the TNF inhibitor to a JAK, we don't have
Yeah.
other biologic data. We don't, and I think the only other study that kind of looked in this area was a tocilizumab versus TNF study, which is a bit smaller and it didn't show any signal of cardiovascular disease or malignancy. But really, this is no other agent has been studied like this as what we'd consider as our second line biologic slash JAK. So I think from this data, it's very clear that TNF should be first. And that's always going to be the way, especially in Europe, because of the biosimilar issue that we're kind
Yeah.
of mandated almost to use it first. But
Anyway. Yeah.
yeah, I don't know really where we are with the second line agents, and maybe it's just as you said, the TNF inhibitors are better, and maybe they're better than every biologic.
Yeah. Well, that trial you mentioned was interesting, and I'm glad you brought it up. That was the Intractate trial, tocilizumab versus etanercept, and because honestly, I don't know what to make of the MACE findings of oral surveillance. I mean, TOFA it was not non-inferior, meaning it was a higher risk. But if you look at the incidence rates of MACE in this trial, it was a lot different than what was seen in the Intractate trial. The etanercept incidence rate was more than double for MACE events in that trial as compared to the adalimumab incidence rates in oral surveillance. So I don't know why it was so different, because that trial was the exact same people, age 50 and above with cardiovascular risk factors and RA. So if you'd seen the same incidence rate in oral surveillance, there would have been no difference between TOFA and the TNF blocker. So you just never know why. To me, it just raises more questions sometimes than answers. Maybe we need to do another trial. The other thing is I haven't not seen the breakdown, and maybe you have the oral surveillance breakdown in terms of comparisons to adalimumab and TOFA versus comparisons between etanercept and TOFA. Have you ever seen things different with adalimumab and etanercept? And we're just lumping them together with all this data, assuming they're the same. But I don't know that I've seen those rates
I don't
stratified for
I don't
MACE
think I've
and the
seen it
other events.
either. I suppose it segregated. I think between North America and Europe, there was very adalimumab in one and etanercept in the other. I think they did show the
Yeah.
breakdown of the geographic data, which didn't seem to show any difference particularly between the regions. but I haven't seen the
Yeah,
individual drug data.
yeah, I haven't either. I'd be interested in that. It'd be good to explore whether there's differences there. And I know with the malignancy, a lot of that was driven by North America as compared to the rest of the world. So I don't know. I certainly think there's areas of further study here, and we could probably just wrap up by saying that the landscape's changed. I think people are being more conservative, rightly so, in terms of who they use these agents in, and trying to apply the lessons learned from the inclusion criteria and the data analysis and the findings from oral surveillance has been really important and a little bit tricky in some cases because I think there's areas of uncertainty. So I would say, look, we got to figure out this VTE thing. I think it's probably real, and it may not be the same with all the compounds, but I guess to me the first thing would be figure out why it happens. That'd be nice. And then the malignancy issue, I don't know. I think that to me was the most troubling finding as we discussed, and I think understanding a biologic plausibility there a bit more, and whether there's differential risk between compounds, I think that'd be really important. So
I think this kind of the bottom line of this trial is a general message, and what it's brought home to a lot of people, I think, is that we need perhaps to be a little bit more cautious in our practice and not be dazzled by the new fancy agent, which is showing some relatively small incremental benefit, and we should pay attention to the vast wealth of like 20 years of safety data we have with our existing agents and weigh that up as being something we should have confidence in and factor in as more important.
yeah, yeah, and I just think more head-to-head studies in the future would be useful, particularly between the JAKs, because again, I'm not. It's not clear to me that we should be lumping these drugs together, at least with some of these events. So I think we need to. I think we need to further examine that. So, and the data we have to date would suggest that we shouldn't be lumping them, to be honest. So, I think that's the future. And of course, there's new types of compounds to come after that.